Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody back to In the Interim. I am your host, Scott Berry, and I'm joined today by my co-host, Kert Viele. Dr. Kert Viele is here, and Kert needs no introduction, as he is a regular on the show. We have an interesting topic, and the topic, I'll introduce the topic a little bit by telling you a true story. And Kert is related to this story. For those that didn't know, Kert and I were fellow graduate students at Carnegie Mellon. And when I was a 23-year-old first-year graduate student at Carnegie Mellon, I called my dad. And of course, the aspect I called him is, I called him and informed him that my wife had taken a pregnancy test and it was positive. And I don't know if many of you in the world have similar interactions with your father when such a thing happens. But
I think the answer is no, Scott.
I think the answer is no. The question, the first thing he said after I said that, by the way, I did say we took the pregnancy test, and the pregnancy test says it's 99% accurate. And I tell him this, and his first question to me was, "What was your prior probability?" Honestly, that's what he asked me. And my response was, and Kert knows this well, I was a married graduate student making a stipend. I said, the test was $15, Dad. The prior probability was pretty high. And his next comment was, "Oh, congratulations, that's fantastic." And he was excited for this whole thing. So the point to that is, if our prior probability is low, the 99% means something different about at the end of the day, the question is Tammy, is my wife pregnant or not? In that, based on this experiment, where the prior probability makes a huge difference. And many of you have probably seen the rare disease examples where if you have a low prior probability of something and you take a diagnostic test and it says positive, you're still probably unlikely to have the disease. So in this circumstance, if I had a 1% chance that she was pregnant and the test was 99% effective, meaning if she's pregnant, 99% chance it says yes. If not, 99% chance it says no. I think they're actually more accurate than that. But if I had a 1% chance, it's only 50-50 she's pregnant in that circumstance. Where if I started with a 10% chance, it was over 90% she's positive. The point to that is the prior probability of that changes my interpretation of the result of the experiment. So the topic for today is using Bayesian borrowing, and that's a case of Bayesian borrowing of what my dad did faced with the result of a clinical trial. And the diagnostic test is a clinical trial. A clinical trial is the same thing. Based with that information, the prior probability was a huge part to his interpretation of the result. There are cases now, and the Bayesian guidance is out, where we use Bayesian probabilities going into the trial to interpret the result of the clinical trial, working with the FDA. And today's topic is doing that. What does it look like? What are the characteristics? What's the behavior? Have we done this before? So that's the topic for today, Kert.
All right. Sounds like a good topic. And it sounds like we're talking about, so we've done this in a couple contexts, and it sounds like you're going to talk about the second one. We've done this in context where we're only borrowing on a clinical trial. There are two arms of a control. There's novel treatments. We've done situations where we've borrowed only on the control arm. So you're comparing to an existing drug. There's lots of data on the existing drug, and you're trying to bring that in. That's one problem you might solve. It sounds like today we're going to talk about the what we'll actually consider the more complex version, kind of a newer version of we're borrowing on both arms, where we have some idea about what the treatment effect might be based on history. And now the question is, what does our current trial have to do in order to, this is a journey from we know nothing in order to we know the drug works. If we're partially there based on what we've already seen, what else do we need to know? How much more evidence do we need to get over that bar? And I know that we've seen this in the context of, completely external trials. We've seen it in the context of, people run one trial and get p equal 0.06. And now the question is, what do we do? So where do you want to head from here, Scott?
Yeah. Good. So first of all, just to, and by the way, this is a shout out to anybody who listens, watches in the interim. If you have questions, throw them our way. We're looking for interesting topics. This was something that somebody contacted me and asked us, have we ever gone to FDA using Bayesian borrowing? And it was a little bit like, whoa, yeah, this is kind of what we do. And I was a little almost surprised by the question, but then it made a ton of sense that now with the Bayesian guidance out, and they were interested in this interaction and what does it look like. The other part to this is the Bayesian guidance makes reference to Bayesian trials where you're controlling type 1 error or you're not controlling type 1 error. And I was asked by somebody else, a brilliant researcher actually, Dr. Bijoy Manan, about, he's a stroke neurologist in Calgary, what's the difference, that sort of thing. So I think we can tackle all of those things today. So maybe we should set it up with a case where this may make sense, or cases where we've done this before, just to give people an example of it. Now, first, maybe we should set up what is done in a frequentist standard clinical trial is you calculate for that trial the probability of a type 1 error. And it's only data in that trial that goes into that calculation. And we say, if the probability of achieving the data in that trial, given the null, is less than 2.5%, we're going to call it a successful trial. By the way, in the circumstance of the pregnancy test, assuming she's not pregnant, the probability of it saying pregnant is 1%. Statistically significant in that case. Now, that might be how we interpret it. Now, here's a circumstance, and it's not uncommon that a company, a sponsor, has run a trial, and the trial is not convincing for approval, but has some positivity to it. Maybe it's a p-value of 0.07. Some cases, it's actually been a p-value of 0.025, but yet FDA is not convinced. Regulators are not convinced. So let's take a case where there's positive data and a p-value of a one-sided 0.04.
And I think, Scott, to this, we should add another situation this happens is somebody runs a phase 2 and gets really positive results, but it's not quite there. And their question is, hey, haven't we already fulfilled at least part of the bar for approval? Same kind of idea. We're partway there, but we're not fully there.
So company A has run a trial that's not statistically significant, but it's close. And maybe they even went to FDA and say, will you approve us? Now, I've never been at the FDA, but I can imagine them thinking hard about it, actually thinking the drug probably works, but it hasn't met the thing. We can't approve it yet. So company A is in that situation. Do you tell them you have to go and run a trial, ignoring all of the things we know from that first trial, and you have to jump a 0.025 hurdle, and we ignore it? Company B has no data, and they have a treatment for the same disease. They want to run the same trial. They're asked to run a trial with a 2.5% type 1 error. Are the companies asked to do exactly the same thing? Because a ton of scientific sense that first company A can utilize the information from that trial, and maybe they don't need as large of a trial to demonstrate that the drug works. So that's a circumstance that we're talking about today, where you have prior probability going into a new trial that the treatment's effective, and you want to use that to analyze the new trial. And it could be that first trial is a phase 2 trial. It could be that first trial, is a biomarker trial that just happened to show clinical benefit. There's a lot of ways in which that information comes about. Another circumstance, and we did a complex innovative design trial where this was a public presentation of a circumstance where a company had demonstrated benefit in 2 types of epilepsy, and they wanted to investigate a third. There's a commonality in the diseases. There's a commonality in the endpoint, the mechanism of the disease. You're taking a treatment that's been shown effective in a related but slightly different disease, and it clearly benefits these other 2, and now you're going into a third. Should the standard be a trial in that subtype of disease, 0.025 type 1 error, or should there be some sense of borrowing from the others? And for example, in this case, it was showing about a 30% reduction in seizures in both of the other types. If all of a sudden, relatively early in this third type, you're seeing a 30% reduction in seizures, you're pretty sure the drug works because of that prior probability. These are cases. The third case talks a lot about it is you might have conclusive proof of benefit in an adult population. There are rare circumstances where pediatrics have the same disease. It's very rare. It's hard to run a trial. Do we just ignore them? You might run a trial and borrow from the adult in some way. So there's a lot of circumstances that you go into a phase 3 confirmatory trial with prior information about the efficacy of the treatment that's in that trial.
And one thing to add to this, these kind of situations, they've come up in the past, and there are often ways that regulators have made the bar a little bit easier given this prior information. And that may be, 1 trial versus 2. It may be, hey, we'll change something about the, what you have to do in your phase 3 trial. What we're really talking about today is trying to formalize this process. So the question is, if you want to apply all the statistical rigor that we usually apply to a trial, ordinarily this is, you got to do 0.025 all the time. Is there a middle ground where we could basically formalize and say, here is how we're going to analyze it? We're still going to keep all the statistical rigor, but we're going to recognize that you do have this, you're partway there.
Okay, so suppose we have this prior probability, and we have a prior distribution about a treatment, and we're going in and we're running a new experiment now. And we're going to collect data in the new experiment. And the Bayesian approach is we're going to use this prior in some way. We'll talk about different priors and that dynamic discounting and those aspects of it. But we're going to use that prior in some way with the new data. Now, explain why that can inflate type 1 error and what type 1 error means and why it's a little bit disconnected in the way we're going about doing this.
So what's going to happen here is suppose that, my whatever my prior evidence says is I think I have a 10% effect, 10% beneficial effect on something. When I come in and compute type 1 error, what I'm saying is let's assume that the control and treatment arms are exactly equal to each other. Tell me what is
the new experiment.
In the new experiment.
Thing. Yep.
Yeah. Yep. So we got our historical evidence on one side, we got our current trial on the other, which I'll refer to it. If there's no effect in the current trial, now this is something we don't think is true. We have prior evidence, so we at least are less likely to think it's true is the more precise way to say it. But if it is true, what's going to happen is that prior evidence, it's going to bias things upward. It's going to say, hey, if I didn't see an effect in the current trial, I'm going to average this in some way with the beneficial effect from the past. I'm going to pull things up a little bit. I'm going to basically say, maybe I have a 2 or 3% effect. The net result of that is that little bit of bias makes you more likely to claim efficacy if, all of this is if, you don't have an effect in the current trial. So the key issue is, what is the value of that assumption? And this is where we get into the guidance and, do we control type 1 error? Do we not? We will often do that calculation and say, if this is true, here is the type 1 error, the results of the type 1 error for various kinds of designs. And they're going to be higher than 2.5% because of this bias from the past. The argument that you make is this assumption that I'm worried about, the null hypothesis is true. I've already partially disproven it. So the value of that type 1 error calculation is less in that case. And that's where we get into, I know where you want to go, discussions of with and without type 1 error.
So if you're going to use that prior distribution to it, and what might that look like is, we're going to use the prior, use the new data, and say we need a 97.5% probability that the treatment's beneficial after the combination of the two, that's going to be successful. We may go to FDA, and that's the criterion. Now, if we weren't using prior distributions, that probably controls type 1 error at 2.5% if we're using a flat prior. That yep. Bayesian analysis is similar to a frequentist analysis. And so the type 1 error would be controlled for the new experiment. And that would satisfy in the guidance where it says you are controlling type 1 error, because really all we're using is the new experiment, because we're not bringing any prior. Once we bring that prior and we combine them together now, as Kert said, to get 97.5% with the combination, the new trial doesn't have to be 97.5 by itself. That might be 93% is enough combined with the prior to say, overall combined we're 97.5. If you evaluate only the new experiment, it might have a 7% type 1 error in that circumstance. And that's where the FDA might agree, okay, the data you're using is reliable, it's relevant, it's appropriate. We agree that you can use that for approval as a combination. Then they're approving within only this modular new experiment the type 1 errors above the traditional 2.5. And maybe it's 7% in that circumstance. So that's kind of the difference between a Bayesian analysis that controls type 1 error for the new modular experiment and one that doesn't, which the guidance talks about a lot. Yep.
We're talking about the combination. I always tend to view this as if I were doing things from scratch and I'm going to do 2 trials, is that combination good enough? And the question is, I'm basically designing my new experiment when the historical data is historical. It's in the past, so I can condition on it.
So let's and we'll leave as a separate topic where we're borrowing only on the control arm. Yes, that can inflate type 1 error. It's a different kind of thing about the new temporal differences in the new trial and others. So we're talking about the circumstance where we're going in with an informative prior about the treatment effect. And you're having discussions with FDA about that.
So I'm going to ask you, I'm going to derail this slightly, and I haven't told you I'm about to ask this, but what about situations, Alzheimer's, sepsis, where nothing has worked in the past? That potentially should give us a negative prior. How should people view that?
I think it's appropriate, and I think it's been an unspoken thing probably at the agency. And there was a time not long ago where there were 25 straight failed phase 3 trials.
Which is unlucky, by the way. They should have won one by chance.
Yes, somebody should have. Yes. And that, by the way, the 26th trial, the FDA may be weary of that if all of a sudden the 26th trial shows p-value of 0.02, maybe they're the lucky one.
Yeah.
And you can imagine a skeptical prior, which is talked about in the guidance in that circumstance. And maybe they even ask for a higher threshold of that circumstance. So that's kind of a different prior going in, where historically the scenario is been nothing works in that scenario. And so you carry the burden of the disease with you as you go into the agency, which I think is quite reasonable.
And you don't want to discourage research in these areas and so on. It's a really hard problem. I wasn't expecting an answer for it, but we should be honest about it exists in the other direction too.
Yep. Okay. So what this may look like in the interaction. So we're in a circumstance, and by the way, we get the challenges of this. So for example, we might get sponsors that come to us that ran a phase 3 trial, and it was a p-value of 0.3, barely observed positive differences. They find a subset of 30% of the population that, wow, it's nearly significant. And they want to take that subset and ignore everything else and go to the FDA and borrow from that subset in a new trial, and it gives them a jump start for approval. And now they've cherry-picked from the previous trial. And by the way, they can explain why, of course, that's the relevant subgroup and all of that, and they want to go to FDA and borrow from that. And there are circumstances where you and I, they come to us, we'd say, if we were at FDA, we wouldn't accept that. Because we believe this is largely about multiplicity, and now you can't pull that out and create a prior that's only based on that. If you want to use a prior based on the entire trial, that might be something to discuss and have that. But to self-select data and only use that data going to the FDA, you're probably going to get a no. And by the way, historically, that's happened to companies, and they come back and say, FDA doesn't do Bayes.
Yeah, that's always frustrating.
Right. It's had nothing to do with Bayes. It's you're misusing it in ways that we as Bayesians would say, no, I would not accept that circumstance. So there's a huge part of the data you're going into the FDA. What are you using? Has it been, self-selected? Is it all of the data out there? Is it relevant for the new trial? So there's a huge part of this, of the science. Science is hard, and you go in. So if we go in with a circumstance where we believe we're bringing to them a relevant prior based on the previous information, that becomes part of the discussion with the FDA. Is this reasonable?
Well, it becomes a major part of the discussion with the FDA. I think we spend as much time on data as we do on the methodology at this point. It certainly is the main reason, as you said, FDA says no. A lot of times we're not told where this subset came from. It's, hey, I want to do a trial in this population, and this is the data I have on this population. And, at some level, that's a red flag at this point that I need to ask the question, you said this is the data we have on this population. What's the data? What's the world actually look like, so that I can assess whether this is it cherry-picked? How much is it cherry-picked? All of these things come into play.
Yeah, the dog that didn't bark, what are you not telling us? Because we get told the rosy part of it, and what are you not telling us, and what data are we not using? And so, for example, in the case of the complex innovative design program, where we've got these two other indications, and they don't have a third indication that failed, for example, if they did, you'd want to include that in a hierarchical model. Or there's a large phase 3, and we're using the entire estimate that comes from that to borrow for the new trial, and we go to the agency, and we're presenting them how we're doing the borrowing. So I'll give you one example of a case that the device was approved. The WATCHMAN device is a left atrial appendage device that's been approved by CDRH at FDA, and they originally ran a trial that was inconclusive. And the name of that trial, the original trial, I'm going to get this wrong. The original trial was Protect AF. And then they ran a second confirmatory trial called PREVAIL. And the PREVAIL trial borrowed where we discounted the first trial by 50% in a static way. We'll just discount it 50%. And by the way, this went through FDA, went through Bram Zuckerman's group. Bram is a brilliant scientist and understands what it means to borrow, the implications for the new trial. More than once, these types of things have gone through the cardiovascular device division at the FDA. Absolutely brilliant scientist who was involved in this particular case in that borrowing. So we go in and we propose that level of borrowing and show what happens in the new trial. The most important thing to show, and I had a client discussion yesterday with the same thing, is what example trials of the new trial, when is it successful with the borrowing, and then how does that compare to if we didn't borrow? So for example, if we've got a time to event analysis where we're borrowing data, if we don't borrow, we need a hazard ratio of 0.7 or better to win. With borrowing, we show that if it's between 0.7 and 0.77, with the borrowing from the historical data that showed 0.7, those are still successful.
The field goal.
Yes, the Bayesian field goal. And so you show them, I think sometimes we revert to operating characteristics. If the truth is 0.7, our trial is 93% powered, we would tell the FDA. Or we would say if the device doesn't work, the new trial has an 8% type 1 error. And we talked about what that means is that additional trials are being demonstrated with the final analysis at the end being Bayesian as a combination of the trials being successful. But I think that's hard for the clinicians at the FDA, that's hard for a proxy for Bram Zuckerman to understand, okay, what am I agreeing to in that? Show them an example of a final trial that the final trial shows an estimate of 0.76. The Bayesian analysis gives a mean of 0.72 and a confidence interval that's less than 1, that this would now be approvable. They can see exactly what they're agreeing to that. So that's one of the things we show them is depending on the result. We also want to show them if the new trial shows a hazard ratio of 1, it doesn't say, oh, the treatment works.
Yeah.
They would not, they would say, wait a minute, that we don't agree to that. We don't want this Bayesian thing to flip that. So what is it flipping?
So we've had a couple situations. I can think of one in oncology with rare cancers where this was like your epilepsy example where we're trying to extend the label. And so we're borrowing across indications. And one of the things the FDA was particularly concerned about is, remember, these are all rare. We may not enroll very many. They never wanted a situation, and I should say ultra rare. I mean, we're going to get a handful. But they didn't want us coming in and saying, we got 0 responses in 4 patients, and we're going to say you should approve on the basis of the prior. we need it to work somewhere, which makes perfect sense. We need some evidence that it's doing something. And so we've had those discussions, show us the minimum that we've had. I remember one a while back, we were borrowing adult data to pediatrics. And again, we were going to get small sample sizes. And the FDA actually said explicitly, we would be willing if you got 15 responses out of, and I don't remember how many kids we were going to be able to enroll, but basically if you can get 15 out of 25, we'll be happy to accept that we think that's enough given the adult data and everything. And so we basically back-solved, what would this do, and then showed that this had reasonable operating characteristics as well. But that was the way the interaction went in terms of, here's the data that we think would be convincing to us.
And so through this interaction, we're showing them the new power. We're showing them the type 1 error of this new modular experiment with the Bayesian analysis that happens at the end. We're showing them example trials. We've had people at the FDA say, okay, your new experiment has a 8% type 1 error rather than 2.5 in that. What if we just say, you can run a new phase 3 trial without Bayesian borrowing, and you get an alpha of 0.08. That we go to that, in many circumstances, that gives the same success and failure of approval that the Bayesian analysis creates at the end of the day.
Okay, you're asking me a trick question. So this one gets a little nuanced here. And I think the answer does depend on, are you borrowing only control? Are you borrowing control and treatment? Are you borrowing dynamically, or are you borrowing statically? I'm gonna certainly focus on the dynamic borrowing, which I think we prefer to do. And I'll have to talk about what that is, because I don't think we've really brought that up. The dynamic borrowing, the idea is if you're walking in with prior information from a previous trial, suppose your current trial is ongoing and it just looks different. So the rates are different than what you saw in history. The treatment affects what you saw in history. The notion is, should I continue to believe this prior, or should I go, hey, something different is going on there? And if you got into the math, what you're really saying is you have a prior where maybe the current trial matches history and maybe it doesn't. There's an aspect of that to your prior. So what happens when things don't match? You would borrow less. So you start to give less weight to history. And this is a mitigation strategy. When you have mismatch, you borrow less, you're less prone to make these false conclusions. Doing that is far better than just increasing the alpha, because increasing the alpha, basically you're saying, I'm always willing to take this extra error regardless of what happens. And you don't have any of these mitigation strategies. So I think it's really important that you go, I'm changing the alpha because I have evidence that it works, and I should be able to discount that evidence appropriately if the new data says it's not right.
The other nice thing that comes, two parts that comes from the Bayesian part that's a little bit different than just saying, oh, you get 0.08 alpha, is that there was an explicit incorporation of the strength of the previous data that generated that 8% was what you got in the new experiment that gave you a final answer of 97.5% or 99, whatever that is. So it's not arbitrarily, oh, we think you get 8%, you get 11%. It's the combined data that tells you what that implied value is. Yeah. But the other part is, if you just take 8% one-sided, your estimate at the end of the day is not utilizing that information. The confidence interval doesn't match the result of the trial in that, and the estimate can be as important as the final result in the trial, which is a combined estimate. So there are, I believe there's lots of advantages of using the Bayesian approach to generate the final combined analysis of all of the data that we know, jumping a regulatory hurdle.
And a lot of times that can be even borrowing good information. You may actually pull the point estimates down. We did that. Actually, it was the same rare cancers trial. Yeah. It had 9 groups in it. One of the early data sets, it's a blockbuster success, but it estimated a 60% rate. And the control was estimated to be about 10. So this is a huge treatment effect. But when we did all the borrowing, it didn't raise the 60%. Our prior, it certainly moved it. It was positive information, but it only estimated it to be about 48 because it pulled it back and said, look, we don't think it's that good. There were also aspects. It was the highest group among many, and we've talked about that in other podcasts. But anyway, what was really cool about that is they continued to do follow-up, and they continued to enroll a few more patients. And lo and behold, the final data came out pretty close to 48%. So, I mean, it will pull things down.
Okay. So we've touched on a number of these issues. I'll give you an example of a case where this happened. Now, we have a number of examples of trials running right now at the FDA where there's been an agreed upon Bayesian analysis of this. We're going to them. I'll give you an example of one where there was this agreement, the trial ran, and actually the data's been publicly disclosed because it went through an advisory committee meeting. So the treatment is called REBYOTA, and it was a Ferring company Rebiotics, and the treatment is for C. diff infection. And this REBYOTA is now approved by CBER in it, and they had run a phase 2 trial. All of this information is public. It's a wonderful thing about the advisory committee meetings is that a huge amount of this is made public, and we're doing less advisory committee meetings now, and this is so a negative of that, a whole separate topic. But so the information's available in the public register on this. So they ran a phase 2 trial, and this is a treatment for recurrent C. difficile infection. This happens when individuals might be treating for cancer or a different disease. They take antibiotics. It kind of destroys the gut biome, and C. diff, which might live in all of us, takes over and now flourishes. And it causes debilitating GI issues, diarrhea, can actually be fatal in it. Now, what they might do is give you another course of antibiotics and hope it sort of cures it. The recurrent aspects is it doesn't. And this is a horrible disease. The treatment is actually a fantastic treatment. It's a fecal matter transplant. They take feces from a healthy gut biome, and through an enema, they give it to the individual. And the hope is that this restores the GI tract, healthy probiotics, healthy gut biome to fight off the C. diff infection. So they ran a phase 2 trial that was not statistically significant, showed a benefit of about 15% in preventing recurrent disease. This is a fantastic trial where there's only one endpoint. Sure, mortality is looked at, but the only endpoint is, does the disease come back? There isn't a vast scale. There's nothing. There's one endpoint in the disease. Does the disease come back? And the proportion of time it comes back. And if it doesn't come back within 2 months, you're in great shape, very unlikely to come back. It's almost a new circumstance of it. But if it does come back, then you're dealing with this case. They ran a phase 2 trial and showed benefit. They're now running a phase 3 trial. It's a rare disease. And interestingly, it became very challenging to enroll this trial because there were outfits using FMT, fecal matter transplant, open biome, for example, that patients could get. And this was not being shut down. The FDA could have shut down all these things, but they decided to, at their discretion, not. but it had never been approved before. So there's a circumstance where there's a belief this is beneficial. This phase 2 trial is showing. Now they're trying to run a phase 3 trial and challenging to get to 0.025 in this circumstance where I think largely the scientific community believes this works. So they agreed to borrow from the phase 2 trial in the phase 3 trial, have a combined analysis of those two trials, meeting 97.5 with the combination of the two trials. The phase 3 trial read out, and it by itself, if you analyze only the phase 3 trial, would have been about 93% probability the treatment works. In and of itself, that experiment didn't jump the 0.025 hurdle. Combined with the phase 2 trial in the pre-specified analysis, pre-specified to the phase 3 trial, but not phase 2, said 99.1% probability the treatment is beneficial when you combine together the two trials. In the way that we worked with the FDA in creating this combined prior with the phase 3 was 99.1, which had jumped the 97.5 hurdle. That's the standard for clinical development. Given the rareness of the disease, it's not a 2-trial hurdle or the hurdle. We don't know whether we're in the 1-trial, 2-trial world of FDA in this circumstance, but given the rare disease, it very much fit into the 1-trial level of evidence there. This went to panel, public disclosure, and of course the interesting thing is the statisticians were a bit of the hurdle as to whether this is appropriate. The statisticians split their vote 1-to-1 for it, but it was voted 13-4 that it demonstrated efficacy in the circumstance. The treatment has been approved by FDA. It's on the market. And an interesting thing about it is that the FDA label for REBYOTA gives the 99.1 that came from the Bayesian analysis. There are no p-values in the FDA label, but it has Bayesian posterior probabilities that it describes the combination of phase 2 and phase 3 as the estimate of efficacy that comes out of that. The treatment effect relative difference estimate is based on that. And interestingly, at the FDA advisory committee meeting, the FDA statistician presented the posterior distribution based on the combined, showed a picture of the posterior and talked about the probability it's above 0, the probability it's at least 5% better, the probability it's at least 7% better. And this was, based on FDA's analysis, the best summary of efficacy we have to present to you advisory committee meeting. And that picture of that stood on the screen for a long time as here's what we're trying to make a decision on today, which was sort of cool. So you can go forward. And they do make reference in the new draft guidance of REBYOTA as a case study of that, and the data's publicly disclosed for your scrutiny.
And the publication's available. It spends more time talking about other aspects of it than the pure Bayesian part. The adaptive design reports in there, the Bayesian models in there, all of the pieces available for it. A lot of our best stuff is in the supplementary materials.
Yes, exactly. I would have loved to have stood up and explained to them all of the Bayesian machinery, but that was not deemed to be the right thing to do in the circumstance. By the way, it'll be another podcast that we talk about that particular example. A lot of Anna McLaughlin, Joe Marion here at Berry Consultants, Lindy Banky at Rebyotics was the clinical lead of this, and the one who spoke to the advisory committee meeting about it. She's fantastic, sort of thing. So lots of people involved in that particular case with it. Okay, so have we missed any parts to what it looks like going to FDA, good circumstances, circumstances, by the way, that we say no to a client. No, we're not going to go present that. We would say no if we were at the FDA. We're not going to present a Bayesian analysis of this. But we've done this probably a couple dozen times over the years, probably more than that. We've been told no by the FDA, by the way. We've presented what we thought was a very reasonable scientific argument for that. We've been told no. We've been told yes. We've been told, can you change it in the following ways? In that, so interaction is not uncommon in those cases.
We've been told to do it when we didn't propose it.
Yeah. The rebiotics, actually, circumstances was a suggestion of the FDA. Bram has made that suggestions. Other scientists at the FDA have suggested, you should explore Bayesian methods in the circumstance. Of course, going back to the original introduction of this, the home pregnancy test is now one of your colleagues. That was Nick Berry, who is now a 34-year-old scientist here at Berry Consultants. So the test.
Thank you for making me feel old, Scott. Appreciate that.
Yes, I know. As you babysat the youngster years ago.
So we should. I think the new podcast, we should always start with a story of you from grad school.
Yeah, we have some of you too, so the turnabout is fair play in all of this, yes. All right, well, we hope you enjoyed this little Bayesian interlude here, which we hope is a more common thing with the FDA guidance. And until next time, we will be here in the interim.
Thanks, Scott.